My name is Laura Vivian, and I'm the President and CEO of Premier Pathology Solutions, a laboratory consulting company. I'm also the Histology Manager for Capital Digestive Care, and it's a large independent laboratory based out of the DC metro area. So we're going to go over the principles of immunohistochemistry, and I'm just going to do a basic overview of specific areas. And I'm going to talk about, so the basics of immunology, we're going to go over some simple terms just with immunology, going over what the terms are that you're most likely going to hear in immunohistochemistry. We're going to go over the antibody types, their characteristics, how the antibodies are derived, and the components of the actual antibodies. We're going to review target antigen methods, the differences between the methods. We're going to look at the structure of the methods and also how the methods react. And lastly, we'll talk about the sample preparation for performing immunohistochemistry testing on specimens in the clinical laboratory. So first, I want to go over what is immunohistochemistry, also known as IHC. And it's a method for localizing specific antigens in tissues or cells based on antigen-antibody recognition. It seeks to exploit the specificity provided by the binding of an antigen and an antibody at a light microscope level. I want to go back and highlight the beginning of the definition. An IHC is a method for localizing specific antigens in tissues or cells based on antigen-antibody recognition. And I really want you to pay attention to the antigen-antibody recognition as we go through the presentation, because that is kind of the base of what immunohistochemistry is. So I'd like to review the general immunology terms that you most likely hear whenever we talk about immunohistochemistry, and we're going to start with antigen. And the actual definition of antigen is a substance that can induce a detectable immune response in animals or cell cultures for the production of antibodies. And we're going to go through what an antigen is exactly within immunohistochemistry and the role it plays as we go further. But just remembering that it can induce a detectable immune response in animals or cell cultures, and it actually produces the antibodies. So going into antibody, we are going to break it down into antibody class and structure, and we're going to go over what specific antibodies there are: polyclonal, monoclonal, and then we're also going to review rabbit monoclonal, which are really common in the clinical laboratory today. We're also going to go over primary and secondary antibodies and as they pertain to the detection systems. So first, let's go over the general definition of an antibody. And an antibody is a host protein produced in response to the presence of foreign molecules, organisms, or other agents in the body. And going back to antigen, so antibodies are produced when an antigen is present. So an antibody, the abbreviation is capital I, small g, in parentheses. And so whenever you're in your clinical laboratory, you actually see this abbreviation, and it stands for immunoglobulin, which is another name for an antibody. So when you're looking at your reagents that you're using on a day-to-day basis, you actually see this on most of your reagents and understand that that means antibody. So let's take a look at the structure of an antibody. And I really, really want to pay attention to this structure, because you're going to see this structure through everything else that we talk about through the presentation. And the antibody is a Y-shaped protein molecule. So an antibody is a protein, and it's composed of both heavy and light chains. And you can actually see at the bottom where you see heavy chains, and towards the top where you see light chains. The heavy chains actually determine the class of antibody that you have. And so commonly you'll see IgG, IgA, IgE, IgD, and IgM. And you'll see these on all of your antibodies in your clinical laboratory. This is actually the subclass of the antibody. And the light chain that you can see on the diagram will always either be kappa or lambda. The upper arms of the Y are the regions of the antibody that bind to the short arms of the specific antigen. And I really want you to pay attention to that, because it's the antigen binding site. And this region of the antibody is known as the epitope. And it is at this site that the antibody attaches to the tissue. So I really want to kind of do another overview of this and kind of understand the structure of an antibody. Also understanding that an antibody may target more than one antigen, but is only specific for one epitope, and understanding the sensitivity and the specificity of antibodies and the types of antibodies you want to choose in your laboratory when you're getting ready to internalize or use specific antibodies for specific antigens. So let's do a brief overview of the diagram again, and just remembering that the heavy chains determine the subclass of the antibody, and the light chain is always either kappa or lambda, and that the binding site is the epitope of the antibody. So we'll talk more about epitopes, and you'll hear that throughout the presentation as well. So let's start reviewing the different types of antibodies, and we're going to start with polyclonal antibodies. And I really want to talk about how it's derived. How do you get a polyclonal antibody? And so I'm going to break it down. Think as if your blood was drawn from your arm, and an IgG, remembering the subclass of the antibody that's determined by the heavy chain, was isolated from your actual blood and injected into a rabbit. Your IgG antibody would act as an antigen and stimulate the rabbit to make anti-human IgG antibody. So your IgG antibody that's extracted from your blood and put into a rabbit is going to have the rabbit create its own immune response and create anti-human IgG antibodies to your IgG antibody, because your IgG antibody is acting as the antigen. The serum from the rabbit would then be used as a polyclonal anti-human IgG antibody reagent. And so we use a lot of polyclonal, monoclonal, and rabbit monoclonal antibodies in the laboratory. Polyclonal is where it started, and they are highly sensitive because it binds to more than one epitope. So polyclonal antibodies tend to bind to more than one epitope. But this generally results in background staining because it's not as selective or specific to one epitope, that we know that it can only attach to one epitope, but a polyclonal is attaching to different ones. So you're going to see a lot of non-specific background staining with polyclonal antibodies. And because they can be derived from not just rabbit, but goat, horse, sheep, and human, the variability of the immune response from one animal to another makes them very difficult to standardize. So your clones will be different, how they react will be different. And so this is where a lot of the inconsistency when immunohistochemistry first came to the surface. It was just really hard to standardize these antibodies, and they had a really hard time kind of finding consistent staining. So going into monoclonal antibodies, and the development of monoclonal antibodies revolutionized the entire science of immunology. And I'm going to go over again how monoclonal, just like polyclonal, how they're derived. And monoclonal antibodies are prepared by injecting mice with an antigen. And the antibody that's produced by the mouse can be produced in unlimited quantities by tissue culture or by transplantation to peritoneal cavities of mice. So although other animals can be used, generally monoclonal antibodies are always derived in mice that are used in the histopathology laboratory. And because they're generally always derived from mice, unlike polyclonal that can be derived from different types of animal species, they're much easier to standardize, and you're going to have more specific staining with them. So it actually is more specific to one epitope. So going back, polyclonal is more sensitive because it binds to more than one epitope. Monoclonal is more specific because it binds to one epitope. And so when you think about picking the different antibodies that you use in your laboratory, you're going to think about the specificity and the sensitivity of the antibodies and the type of antigen that you're trying to detect. So you probably ask, why would you use polyclonal then at all if monoclonal is a lot easier to standardize, and they're just much more easier to standardize and have more population of the same clone? Polyclonal is definitely more sensitive, and you'll find that your polyclonal antibodies are used again for your more harder to detect antigens. So an example would be TDT in a laboratory. Generally, your TDT is a polyclonal antibody. It's more sensitive. It's going to pick up that TDT antigen within the tissue. And monoclonal is going to be more specific, so it's usually used for your general immunohistochemistry stains within the laboratory. So going on to rabbit monoclonal or mouse polyclonal antibodies, and this is the newest antibodies that are being introduced into the laboratory. This is a new wave, and the technique to develop these antibodies is very similar to monoclonal production, but it's actually the mice is actually infused with rabbit cells. So the whole purpose of rabbit monoclonal antibodies was to get both the sensitivity of a rabbit antibody and the specificity of targeting a single epitope like a monoclonal antibody. And you'll see a lot of antibodies now being rabbit monoclonal, and another way you'll see it is mouse polyclonal. They're becoming very common in the clinical laboratory. So I want to start going over primary antibodies and secondary antibodies and what they are. And Primary antibodies are used within different detection systems. So we're kind of getting into more of the detection and how the reaction starts working. So now that we've gone over what an antigen is, what an antibody is, the structure of an antibody, we're going to start going into exactly how you perform immunohistochemistry. So I want to really touch on primary and secondary antibodies because these are themes that you're going to see in how the actual antigen is detected. So your primary antibodies, they're developed against an antigenic target of interest. So your primary antibody is your CD3s, your CD4s, your CD5s. That's the antibody that you're using within your clinical laboratory. Those are known as primary antibodies. And they're typically unconjugated, which means that they're not labeled. So they're used in combination with secondary antibodies. And again, we'll go through this as we go through the detection. I just really want you to remember primary and secondary antibodies and what they are as you start seeing the detection system. And a primary antibody can be a protein peptide, a carbohydrate, or other small molecule. And they recognize and bind with high affinity and specificity to unique epitopes across a broad spectrum of biomolecules, basically just saying they're going to bind to what they're actually targeting within your tissue antigen. And they can be very useful for the detection of biomarkers in many diseases, which is why immunohistochemistry is becoming a very high volume in a lot of laboratories, and it's a lot of pathologists are using immunohistochemistry to determine these certain biomarkers within tissue specimens. Going over secondary antibodies, the secondary antibody binds to the primary antibody, and this is your typically your labeled antibody. It's conjugated, so unconjugated means unlabeled, conjugated means labeled, and they bind to the primary antibodies to assist in detection, sorting, and purification of target antigen. And the second antibody must have specificity for the antibody species and isotype of the primary antibody being used. So they have to be the same species. So if your primary antibody is monoclonal, your secondary antibody has to be monoclonal as well. It cannot be separate. It won't work that way. So now that we've covered our general immunology, we're going to start getting into the process of detection systems and also epitope retrieval and how this reaction starts happening. So we know what the antigen is, we know what an antibody is, we have an idea of what primary and secondary antibodies, what they are. So remembering that the epitope is the area where the antigen binds with the tissue when we looked at the structure of the antibody. When you do formalin fixation on tissue, there are actually hydrogen bonds that are formed over these epitopes. And they actually, they kind of mask the ability to do immunohistochemistry staining. So you have to open these epitopes to accept the antibody being applied to the tissue. So we're going to go over heat-induced epitope retrieval, enzyme-induced epitope retrieval, and the combination of heat and enzyme-induced epitope retrieval. So the advantages of epitope retrieval is that you have more uniform staining, you have decreased background staining, and you have day-to-day consistency of stains, and it helps you increase the standardization. And as we go through what epitope retrieval is, just remember what the advantages are and understanding how that works with immunohistochemistry. So we're going to start with the components of heat-induced epitope retrieval. And heat-induced epitope retrieval uses heat to open the epitope along with the retrieval solution. So the major factors that govern the effectiveness of retrieval solution or of the retrieval with heat is the pH of the retrieval solution, the volume of the fluid, and the heating time and temperature. And it's really not known why and how the heating method works, but it's really you'll see mostly heat-induced epitope retrieval being used in the clinical laboratory. So I want to go over what the two most common retrieval solutions are being used in heat retrieval, epitope retrieval solution is sodium citrate buffer and EDTA. So I want to go back. Whenever you have an antibody and you're getting ready to bring that into your laboratory and you do research on your on the actual antibody, it'll always have a specification sheet, that will specifically tell you what type of retrieval you should be using for this antibody. And within the heat-induced epitope retrieval, a lot of these are commercially made. So if you're doing immunohistochemistry automated, your antigen retrieval solution is something that you're purchasing. Generally, people don't really make it by hand too much anymore. But to give you an idea of how this reaction works automated is you just think about if you were doing it non-automated. So kind of going through the process, you're always going to deparafinize your slides. And then you're going to do your epitope retrieval, and we're talking about heat-induced epitope retrieval. You want to open your epitopes to be able to accept the antibody being, applied to your tissue. And you would actually put your tissue section in a solution and heat it to a certain temperature, and that'll allow the epitope to open up. So whenever we talk about fixation, and I know that I discussed that it creates hydrogen bonds, I don't know if any of you have heard how fixation is really, really important in immunohistochemistry, and we'll touch on that again whenever we talk about how to prepare your samples for immunohistochemistry. But because we're talking about how to, retrieve your epitopes, if the tissue is overfixed, it's harder to break down those hydrogen bonds and can lead to false negative and false positive staining. So, and the same applies to underfixed tissue. So just remember that that's what's happening. You have to open your epitopes in order to start the process of immunohistochemistry staining. So also we have enzyme-induced epitope retrieval. So we have heat-induced and enzyme-induced. And in this method, an enzyme is used to expose epitope sites. So in the last method, we talked that heat in a retrieval solution opens the epitopes. In this method, we're talking about that the enzyme opens the epitope. The enzyme digestion is an older method of the two, and it's used a little bit less frequently since the introduction of the heat-induced epitope enhancement, but it is still used in the laboratory. And generally, again, just like if you're doing automated with the heat and you're purchasing that solution, same with the enzyme. You're probably purchasing a commercially made enzyme to be used with your antigen retrieval and your immunohistochemistry staining. And there are a lot of different various enzymes that can be used. And again, generally they're commercially made. But knowing the concentration of your enzyme is very, very important because enzymes can increase non-specific staining, but more importantly, they can actually break down your tissue and cause tissue fragmentation. And so when you buy it commercially made, generally your vendors will make usually a 3-tier type enzyme, like a 1, 2, and 3, meaning the weakest, moderate, and the strongest enzyme. And knowing what enzyme works best with that antibody is really important, or you will have tissue distortion from over-digestion. Going into heat-induced and epitope-induced retrieval combined, this is in part due to the enzyme method alone for epitope retrieval can actually reduce the quality of the tissue morphology if it's over-digested. So sometimes you'll see heat and enzyme combined, and this way it kind of minimizes that damage that happens from the enzyme. So usually how it works is the heat is generally performed first and then followed by a rinsing, and then the enzyme is applied for a short time. This retrieval for epitope retrieval is less common. You don't see it so much used in laboratory. Generally you're going to see heat-induced and enzyme-induced. And going back to the enzyme, just so you can correlate that with what you're using in your clinical laboratory, is mostly what I see when I'm performing immunohistochemistry is your enzyme will be labeled as a protease. So if you're using any reagents on your carousel or in your automated system and you see something that says protease, that's your actual enzyme that you're using to detect your epitope. So now that we've talked about the retrieval of epitopes, we're going to start talking about the detection systems. And whether manually or automated, once the tissue is deparaffinized and you perform epitope retrieval, these are the steps. You always deparaffinise, then you open your epitopes. Then the detection systems start being placed on the tissue sections to create the reaction between the antigen and the antibody and allow visualization of this reaction. So we're going to go over different specific detection systems. The first one being the direct method, secondly being indirect, the third being avidin biotin complex method, also known as ABC. And we're also going to go over a labeled streptavidin biotin method, also known as LSAB. And we're lastly going to go over polymeric detection system, which is what most of us are using in our laboratory today. So going over the direct method, this is a one-step method, and a labeled antibody of known specificity is used to identify antigens in patient's tissue. So it's you attach a label by chemical means to an antibody and then directly apply this labeled conjugated antibody to tissue sections. So this method is very simple and it's very rapid. And generally you use this method in direct immunofluorescence. So if anyone's performing direct immunofluorescence automated in their laboratory, you're using a direct method. And it's labeled with fluorescence at the end that's usually viewed by a fluorescent microscope. So I want to take a look at a model, like a structure of a direct method. So looking at the model, you can see at the bottom, it shows your tissue antigen, which is your patient tissue, your primary antibody, and your label. Is very simple. Your primary antibody, your label being what's visualizing that you can actually see that reaction happening. So there's not too many steps in there in between. And because it's very minimal as far as the reaction, the sensitivity is low due to the little signal amplification that's being provided by the process. So we're going to go on to the indirect method. And if you look at the steps in this method, you can see that something's been added. And you can see now that we're seeing the primary and secondary antibodies that we went over in the definitions. And so you're going to see primary and second antibody actually going through the rest of the detection system. In the indirect method, it is a really simple modification of the direct method. So again, I'm going to go through the steps just so everyone can kind of keep in mind of how this happens. Always deparaffinize your slides. You're going to retrieve your epitope by either heat-induced or enzyme-induced, or both heat and enzyme-induced. And then in this detection, you're going to apply your primary antibody, which is the antibody that you're trying to detect. It has to have the specificity against the antigen in question and is added to this section. So if you're trying to detect a CD3 or CD4, this is your primary antibody. The labeled secondary antibody, which has the specificity against the antigenic determinant of the primary antibody, meaning it's the same class, is then added. At the end, you can see where it has a label of peroxide, alkaline phosphatase, and glucose oxidase. This is your enzyme that's applied. It actually binds your primary antigen and your enzyme to actually visualize the actual reaction. So let me break that down a little bit more. The secondary antibody serves as a label, as it actually connects. It has to be the same species as the primary antibody to label at the end. So the secondary antibody links the enzyme and the primary antibody for it to be visualized at the end. And I hope that makes sense. I kind of want to be as simple as I can with this because it's going to get a little bit more detailed as we go on. So let's go ahead and take a look at the actual structure. So here you can see where the tissue antigen is, the primary antibody. At the top, your peroxidase antiperoxidase complex solution. That's your enzyme. So your primary antibody and your enzyme, your secondary antibody is what links them together so that they can actually form a visualization method to be able to see the reaction. So you're probably wondering, well, what's the benefits of doing indirect versus direct? And indirect method is more sensitive because of the signal amplification that we're adding the enzyme where the secondary antibody links them together. So the next method is the avidin-biotin complex method, also known as the ABC method. And you can see that there, again, are more steps. So as we're going through the detection systems, remember that it started with the direct. We went on to indirect because it was a little bit more sensitive. Because in the clinical laboratory, we're always trying to get things to be more sensitive, more specific, less background stain, and just kind of an overall better immunohistochemistry stain. So as we're evolving, these are the different detections that arose through the history of immunohistochemistry. So the ABC method, you can see that you have, again, your unlabeled primary antibody. You can see that you have a biotinylated secondary antibody, which is your linking reagent to your avidin-biotin enzyme complex. That's your enzyme. And your substrate chromogen, again, is what will be applied for visualization. And we'll talk about visualization once we go through the detection system. This is a very standard immunohistochemistry detection system that was developed. Because biotin binds to avidin with an extremely high affinity and fast on is very high specificity, that's where the avidin-biotin enzyme comes into place because it actually binds and becomes very sensitive for the antibody. It makes the reaction much more sensitive. However, the biotin attached with avidin can produce nonspecific staining due to the high affinity. So generally with these types of methods, you always have to have a protein blocker because you are picking up signal on non-specific epitopes. Because remember, an antibody can only bind to one epitope, so you need that one specific signal that you're looking for. If you're doing this in the clinical laboratory, if you look at a slide underneath the scope and you see that there is, different non-specific staining kind of surrounding the cells that you're trying to target, this is exactly, this is what I'm talking about with non-specific staining. It should only be very clean in the background, specific for one target cell. So because avidin and biotin created the non-specific staining, and you had to add another blocker to, to kind of prohibit that non-specific staining, another detection kit was developed to kind of minimize the non-specific staining. I'm sorry, I skipped over the actual structure. I apologize. So we can actually look at the structure of avidin and biotin. The bottom is the tissue antigen, and you can see where the primary antibody is applied, and then the biotinylated secondary antibody, which is what we talked about, that links the avidin-biotin complex to the primary antibody. So that's the actual structure of the detection system. So we'll move on to the next slide, which is the labeled streptavidin-biotin method, also known as LSAB. And here you can see that you have another step with your enzyme, and you can see that I've actually separated what type of enzyme those are out, and you can see that it says that, again, the first level is unlabeled primary antibody, and the second is the biotinylated secondary antibody, again, just like the avidin-biotin complex. The difference is here is you have an enzyme, and you have streptavidin conjugates, so the horseradish peroxidase and the alkaline phosphatase conjugates are used in here. And the difference between that is it kind of decreases the non-specific staining because streptavidin is less likely to have that non-specific staining like the actual avidin-biotin method. So this method is a lot more sensitive, probably about 5 to 10 times more sensitive than the ABC method. And it was used to decrease the non-specific staining that can lead to false negatives or positives. So this was this was very common in laboratories that they were using this detection system, still with different biotin blockers because you do still have that biotin present in the detection system. So you still had the steps being used to block that non-specific staining just like you did in the avidin-biotin complex. So we're going to take a look at the structure of the antibody, or of the detection system, I apologize. And here again, just like all of the other diagrams, you can see the tissue antigen, and then the primary antibody, which is the Y that we reviewed at the beginning, the biotinylated secondary antibody, and the streptavidin enzyme complex, remembering that the secondary antibody binds the primary antibody in the enzyme complex so that it can be visualized underneath the light microscope. So we're going to move on to polymeric methods, and I really want to spend time here because this is what most of us are using in our laboratories today. The other methods kind of have the LSAB you may see in some laboratories, but generally you're seeing the polymeric used in most of our laboratories. And although many of the streptavidin and biotin methods are still widespread use, like I said, there are certain limitations characteristic of these methods with the endogenous biotin that we need to create blocking biotin steps, which makes the process a little bit longer. And here you can see that the steps have decreased, and it's just a very shorter process and actually increased turnaround time and sensitivity with our detection system. So going through this, with the primary antibody, you can see the enzyme-labeled polymer and the substance chromogen. And in this specific model, and I'm going to flip it so you can actually see the diagram, the HRP and the AP are your enzymes. They've actually been fused with your secondary antibody. So in the other detection systems where we saw that these were separate steps, this now became one step. And so that's where your decreased turnaround time comes from. And biotin has been completely removed from this detection method. So I'm going to go ahead and flip to the next slide. Okay, so looking at the diagram, you can see that there's a unique chemistry here that permits binding of a large number of enzyme molecules, the horseradish peroxidase and the alkaline phosphatase are enzymes, to a secondary antibody via the dextrin backbone. And you can see the dextrin backbone that's there. So how this works is you have your antigen, and then you have your antibody that's applied, and then your secondary antibody and your enzyme are combined. So that's one reagent. And then they react and your chromogen is applied. So I'm going to go over this a little bit more in detail here because this is this is what most people are using in their laboratory, like I said. So again, you're going to deparaffinize your slides, and you're going to do epitope retrieval by heat-induced, enzyme-induced, or heat and enzyme-induced epitope retrieval. And then you're going to apply your primary antibody, which is whatever antibody you're trying to detect, CD3, CD4, CD5. Your secondary antibody and your enzyme at this point are going to be combined. So in the previous methods, we would see that the secondary antibody was applied and then the enzyme to link the enzyme and the primary antibody together. Here it's a combined. So the last step, which is a three-step method, is your chromogen for your visualization of the actual antibody. And we're going to go on to the visualization methods because I know I've been talking about how to visualize the antibody and the label in the antibody and understanding how that reaction goes. So we're going to move on to the visualization of the actual antibody in the tissue. So with the visualization method, is an enzyme in the presence of a substrate and a chromogen provides the indicator system to visualize the location of the antibody. So moving on to the next slide. So keep in mind the enzyme is your horseradish peroxidase and your alkaline phosphatase that we talked about in the polymeric detection system. So I want to talk a little bit about horseradish peroxidase. And this is what, again, most of us use in our laboratories today. It kind of serves as a workhorse in a clinical lab because it's consistent when producing large quantities of slides. And the HRP in the presence of hydrogen peroxide, which is the substrate. So with the horseradish peroxidase, your hydrogen peroxide acts as the substrate. The enzyme is fused with the secondary antibody, and the substrate of the hydrogen peroxide and the chromogen react to create the color that you actually see whenever you perform your stain. And this will actually identify the site of the antibody binding by forming a colored compound such as DAB and AEC, DAB being the brown color that you see, AEC being the red color that some of us use in our laboratories. Again, I'll go over that in the next slide. But just to give you a breakdown of how this works. So whenever you have your tissue section and you cut it, you're going to deparaffinize your slide. You're going to perform epitope retrieval. If it's being done manually, this is something that's being done manually on your instrument, either by heat or enzyme or by both heat and enzyme. And then your detection system is applied. So when we're loading our slides onto our immunohistochemistry instruments, keep in mind that these are the steps that are happening, and understanding what those components are of your automated immunohistochemistry system and how they're actually working to react with the antibodies. So within the detection system, going to the polymeric, because I know that's what most of us use, we have our primary antibody, our secondary antibody that's fused with our enzyme, which is the horseradish peroxidase, here being HRP, and then that also reacts with the substrate, which is your hydrogen peroxide, and then your hydrogen peroxide in combination with your chromogen, which is your DAB, and I'll just talk about DAB now, actually forms your color. So moving on to the next slide, we're going to talk about the DAB and the AEC. So I wanted this slide up because I'm commenting on the DAB and the AEC, and I'll go back to that with the color. But I also want to talk about the alkaline phosphatase enzyme, and it's actually demonstrated by the use of naphthol AS phosphate as the substrate and chromogens such as fast red violet. The alkaline phosphatase, we don't really use that too much in the laboratory because it's very sensitive to heat and light, and it can lead to inconsistent staining. So generally, in most of your detection systems, you're going to see the horseradish peroxidase. And the horseradish peroxidase, with the slide that's up now, can actually react with a chromogen of DAB or AEC. And you can see on the slide where I have that DAB forms the brown to black color. You can actually mount this with synthetic resin, which is your per-mount. You're going to clear your slides just like you would normally do and mount them with per-mount in your cover slip. AEC forms a red color, and it actually has to be formed with an aqueous mounting media, which means that you do not run these slides down through your alcohols and xylenes because it will actually take the red stain out. Generally, when you do a chromogen with AEC, you let the slides air dry and then cover slip them after they've air dried. What's the difference between DAB and AEC? Most of your general immunohistochemistry stains will be done by DAB. Melanoma markers for dermatopathologists are very commonly done with AEC because it gives a red stain with that antigen and antibody, binding. Because if there's melanin pigment present in the tissue, which is already brown, and you actually use DAB as your visualization method, it's going to be very hard to determine if that's truly positive or negative. So on many times, like your S100, your Melan-A, MART1, and your HMB45 will be done using the AEC. Also going into dual stains, and dual stains are becoming a very common part of immunohistochemistry these days, where we're doing multiple antibodies on one tissue section. you're using your two separate visualization kits to detect these antibodies on one tissue section. So you're using your DAB detection system and you're using your AEC detection system, so that when your reaction has happened and the epitopes are retrieved, your antibodies are applied. One is going to visualize with the brown to black DAB, and one will visualize with the red, which is the AEC. Okay, going to the next slide. I really want to lastly talk about the tissue handling of immunohistochemical techniques. And I'm going to go through specific each individual area. So going to the next slide. First, we're going to talk about fixation. And I touched on this a little bit when we talked about epitope retrieval methods. And we really want to make sure that we have complete fixation before processing, having complete fixation before processing, let me rephrase that, is very necessary to achieve optimal results. And we know that fixation and processing are two completely different things. Fixation is whenever your tissue is being fixed prior to being put onto the tissue processor. And the most common fixative in the diagnostic laboratory, as we know, is 10% neutral buffered formalin. The time of fixation is very, very important, like I explained with epitope retrieval. Hydrogen bonds are formed onto these antibody sites, and we have to unmask those for the reaction to happen. And some things that you can see to tell that there's fixation issues. If you have a tissue and in the center it's lightly stained, but on the outsides of the tissue, the reaction with the detection system, and you can see the DAB and the cells are very clearly defined, but in the center of the tissue it's very foggy and you can't really tell. That's lack of fixation. And that can actually really cause false negatives or false positive staining. Sometimes if tissue's underfixed, it won't stain at all, which is why there's very strict guidelines that are out by CAP-ASCO for the detection of ERP or HER2 markers when it comes to predictive testing for breast cancer. Very largely in part to do with how important fixation is with immunohistochemistry. I cannot say that enough. I've actually personally seen when things have not been fixed properly and how that can negatively affect immunohistochemistry or staining. So going to the next slide. So the next slide is processing. And with processing, we really want to make sure that our paraffin in our processors are at their, they should have low melting points and shouldn't be really high, because if it infiltrates way too fast, that can also cause inconsistent immunohistochemistry staining. And again, you can see tissue exposed to high temperature can accelerate damage, again leading to falsely, negative and positive cases. And also, I really want to, with breast tissue, and I want to talk about this because I've worked in laboratories that have breast tissue. Sometimes they don't process properly. We know that that tissue is made of fat. We'll go back and reprocess it sometimes because maybe we think that that'll help the morphology of the tissue. Maybe the pathologist has requested that. In the event that this happens in your laboratory, please be aware that reprocessing can affect immunohistochemistry staining very much so, especially when doing predictive markers and any other antibody, of course. But when doing predictive markers on breast tissue, because that's a lot, breast tissue generally gets reprocessed sometimes often in a histopathology laboratory. We want to be really careful when we're reprocessing tissue and just kind of making sure that our pathologists know that this is what we've done, so that way they can determine, the reactivity of the antigen and the antibody within the immunohistochemistry stain. So going to the next slide. We're going to talk about microtomy because the sections that you take are really, really important to immunohistochemistry too, of course. Your water bath should always be clean and just contain DI water, not tap water, because we don't know the pH of the tap water. So generally just using DI water. No bath adhesives. I know some laboratories do put adhesives in their water baths to help the slides stick onto, or to help the tissue sections stick onto the slides. That can affect or alter immunohistochemistry staining. Positively charged slides should be used. Make sure you have a quality positively charged slide. I've seen not quality positively charged slides being used in immunohistochemistry, and I can tell you off of experience that can also affect the quality of immunohistochemistry staining, especially if you're having issues within your laboratory. A lot of these things that I'm going over with fixation, processing, and microtomy, very important whenever you talk about troubleshooting and trying to find out why isn't something reacting properly. Why is my stain not as dark or as light, or is not staining the correct antigen sites that I would have expected it to. Also, to keep in mind, your section should be free of folds and wrinkles. If your section is wrinkled or folded, that may be the site that the pathologist is actually expecting the immunohistochemistry stain. That's the site that they're looking for to be positive or negative. I've seen it happen many, many times when a pathologist will come back and say, there's only this very small spot and there's a fold over it, so I need you to recut it. And when it gets recut, that spot is no longer there. And so for quality of patient care and quality of what we're doing, the types of sections you take for immunohistochemistry is very, very important. I put two pictures of immunohistochemical stains up here, one being a CD20 stain, so you can see that it's stained with DAB. Your background, which is your counter stain, is always hematoxin and bluing because we just want the specific sites to be stained, correct? So we want a contrast, which is your hematoxin bluing. It's what's used in every automated immunohistochemistry system. So you see the CD20 on the left and your MART1 melanate on the right with the AEC detection. So going back to the detection systems that we talked about, this is an example. Your MART1 melanate, which is a melanoma marker, is being used with your AEC, which is your chromogen for that detection system. Your CD20 is being used with DAB. I just want to go over a summary of what we talked about today, and it's very important to understand the basics of immunology, the reagents, the methods to produce and achieve reproducible, high-quality patient care. And I cannot say this enough, understanding what components you're using whenever you're doing immunohistochemistry and understanding the high complexity testing that you're performing is really, really important. I've gone into laboratories where some people have had no idea what they were doing or what their reagents actually meant to immunohistochemistry. And so trying to understand a lot of these terms that I went over, if you go back into your laboratory after this presentation and you look at some of the abbreviations that we talked about, the horseradish peroxidase, the alkaline phosphatase, the DAB, the AEC, the monoclonal, polyclonal, rabbit monoclonal, the IG, the subclasses of the antibodies, the IgE, IgD, IgM, you're going to see all of these terms in all of your reagents with immunohistochemistry. It's very, very important to understand, even when you're doing automated immunohistochemistry, what these reagents are actually doing while it's being performed automated. Because often as we do automated any testing, it becomes monotonous where you feel like you're just putting slides on and you're pushing some buttons. But it's very, very important for the patient for you to know what you're doing, and especially when troubleshooting in areas that you're just not sure what's going on. If you understand the steps and understand how the reaction is taking place, it's very easy for you to go back and determine what's not happening properly to be able to fix it. So I want to open the floor for any questions that anyone might have.
Thank you, Laura. That was a great presentation, very in-depth. You gave a lot of information, and I just know that our attendees, our participants, have just gained a wealth of knowledge today. We do have some questions coming in, so during the presentation, if you wrote down any questions, please type those into the question box, and we will go ahead and ask those questions. As always, we have a featured product, and this month's featured product is the TissueTech QuickRay, and it's for building TMAs, tissue microarrays, which is basically a collection of multiple cores, and this is great for controls because you're able to put all of these different tissue samples into one tissue block, and you can stain the slide, and there's an example of the stained slides, and you can actually view all of these different tissue samples on one particular slide. If you need more information on the QuickRay, you can get that on our website at sakura-americas.com. And at this time, I would like to ask the questions that have came in. So the first question is, what are your recommendations for IHC testing tissue 6 in GTF fixative? I'm sorry.
Okay, I apologize.
You can always get back to and answer this question offline. If you need to pull up some notes or do some research on this, that's not a problem. I can send you the question directly.
Yeah, I apologize. I was really trying to Google it really quickly, but I can't. GTF, I'm really not familiar with that fixative too much, but I'm really interested in kind of learning a little bit more about it and what specimens are actually being fixed in GTF. So I can definitely look into it. If you want to give me the information, I can get back to them. But just off the top of my head, and I just want to kind of, with fixatives, and some specimens are fixed in Zeus and Michelle's. And so I just kind of want to look, because some of those specimens cannot have immunohistochemistry performed, which is why we do certain things like direct immunofluorescence on specimens that come in a Michelle's fixative. And so if I could just have a little bit of time to review that, so I can give the appropriate answer, that would be great.
Perfect. I'll go ahead and send you the question offline then. Another question is, what is the recommended melting point of paraffin for processing? And, well, you can answer that and then I'll ask the second point of the question.
Okay, generally your melting point should be between 60 and 62 degrees for your paraffin.
Okay. And then the second part of the question is, when using A, E as in Edward, C as in cat, chromogen, can the slide be briefly put on an automated cover slipper and exposed to xylene?
A lot of the AEC methods have evolved from when they first started. When they first started, they truly needed to be mounted in aqueous mounting solution, air-dried and non-synthetic like per-mount. However, the more developed AEC methods, generally what you want to do is you want to stain your slides, you want to let them air dry, and you can dip them into xylene and then cover slip them with your normal per-mount, or you could actually air dry them and put them in a rack and then put them on your automated cover slipper.
Great. Thank you. No more questions have came in, so once again, I would like to thank you for your time. Like I said before, this was a very in-depth presentation with a lot of knowledge, and I do appreciate the time you took to put this presentation together. I did just receive 2 more questions, so I'm going to go ahead and ask those because we do have a little bit of time left. You talked about the time of fixation and mentioned, what about leaving tissues in formalin for a long time? Do epitopes get more difficult to retrieve?
Yes, yes they do. That's why sometimes, I'm going to use tonsil as an example. Sometimes we let tonsil sit in formalin because tonsil is a very common control for a lot of immunos. Most of your CD markers are used for tonsil. It's very often that I encounter that once you stain the tonsil for some of these CD markers, it's dead negative, and that can truly be due to overfixation because the epitope retrieval just cannot open those epitopes to accept the antibody. So absolutely, yes, it can affect your immunohistochemistry staining. And I know that most of us have tissue, control tissue kind of stored in formalin, including myself, and at my lab we do the same. I highly recommend anytime you go to put a new control into place, you have to test it first because there's a very high possibility that that epitope will not be retrieved for that antibody.
Thank you. The next question is, for doing a MART1 stain, is it better to use the AEC detection system than the DAB? I prefer having both available in my laboratory. I like the DAB and the AEC. Dermatopathologists generally like the AEC, the red stain, again, because of the melanin pigment, but I've also worked with dermatopathologists that sometimes have a melanoma case and they'll request that it be done on DAB. So I don't think there's one better or worse than the other, but having both available in your laboratory is really, really beneficial. Perfect. Another question is, any recommendations for performing IHC on tissue blocks several years old?
Again, going back to the stability of the epitopes when something's been paraffin embedded, whenever you do I'll just go back to whenever I do a validation study and I'm bringing in an antibody into my laboratory. Generally, you go back cases, and sometimes it's 7-8 years. Even pathologists sometimes will actually request blocks to be pulled for old cases that they're looking for. You don't ever really want to increase your epitope retrieval too much when you're doing these blocks because it can actually distort your tissue. But there's a possibility that the antigen may not be retrieved because of the hydrogen bonds creating that kind of block for the epitope to be opened up. So really you're going to perform them just like you would perform other immunohistochemistry stains, but running a known control with your tissue that you know works very well for this antibody is really, really important no matter what type of tissue, what type of fixation That way that's your gauge to say, yes, it's working, or no, it's not.
Great. Thank you. Another question is it more beneficial to have controls cut fresh or have them stored?
That's a great question. Most laboratories are going to have them pre-cut, but ones that have been cut for 1 or 2 years, generally you shouldn't use those for controls. If you're going to pre-cut them, they should maybe be pre-cut maybe a week or two ahead of time, but not actually cutting a ton of slides and letting them sit for a long period of time. And that just kind of goes off of any type of, airborne particles that can be put on. I mean, you shed epithelial cells that can be picked up by immunohistochemistry stains, and any kind of just debris that may attach to that slide can actually cause immunohistochemical stains to pick up on that, and can actually cause just for really non-specific staining in different areas that you're just not really too sure what that is.
Perfect. I have 2 more questions, and I think that might be all for today. One question is, we are preparing to begin doing the MART-1 in our Mohs lab. Any suggestions or recommendations?
MART-1, Melon A, is really, it's a great, you retrieve that antibody by heat-induced epitope retrieval. So you want to do it that way. And because it's a Mohs lab, I would probably maybe start with the red counter stain, because you're working with dermatopathologists and there may be melanin pigment in the skin biopsies that you're taking. So starting with the red, but also remembering to make sure you air dry it, because I've actually had that happen where the techs will forget to air dry it and run it down, and the stain can sometimes come completely out. So doing it in your Mohs lab, I'd probably start with the red if you're determining between red and DAB, and then doing your heat-induced epitope retrieval. Depending on what vendor you're choosing to do this test, also really read the literature. I cannot say that enough. And understanding what they're suggesting is the best way to retrieve this antibody from your tissue specimen is really, really important.
Great, thank you. If you have frozen sections, is antigen retrieval necessary?
It depends if they're doing, what type of method, if they're running it on a conventional immunohistochemistry instrument or non-conventional. So a frozen section and how they're fixing it, I guess, would be my question.
And just one more question is, how critical is it to use ID water versus tap water?
It's really critical. I went through a phase where I wasn't too sure. I kind of did a study on my own, because it was like, if you put tap water in your water bath, how will that directly affect immunohistochemistry staining? But each city and location that you're in, the tap water has a different type of pH. And remember that all of your reagents for immunohistochemistry kind of keep a stable pH. And so using DI water, you can determine that you have a consistent pH, so that that's not going to interfere with the reaction between the antigen and antibody. So it's very, very important to use DI water. That's probably one of the most important places to use DI water in immunohistochemistry, along with making your bulk reagents. And Alicia, I want to go back to the frozen section question, because you really can't perform immunohistochemistry in frozen sections, so I really don't want to sound too confused with that question. So I'm not sure if they're doing direct immunofluorescence with their frozen sections or what they're using with them, but you have to fix the tissue in order to do immunohistochemical staining.
Okay, thank you. Well, again, thank you for your time. We are out of time today. I just want to remind all the attendees that the web link for this presentation is located on the email invitation that was sent to you. That is the email invitation that was sent to log on to today's webinar. It has the web link on there. So please scroll down and actually click on the link, and you'll be able to download the presentation. If you ever need to come back to this presentation and hear this webinar again, it will be uploaded to our website under IHC in the webinar library, so you can always come to this for a refresher, or if you wanted to recall something that Laura stated. And once again, thank everybody for participating, and Laura, again, thank you for your time.
No, thank you for everyone. Thank you, Alicia. I really appreciate it. Thank you very much.